fairly small budget project, so we didn't have money to really experiment with new technologies. So we used some like existing live stream cameras that we had. It didn't work like super great to get pollinators. It was just like wind would just trigger the camera all the time. But yeah, we have done some projects that are more specific, kind of using cameras to monitor stuff. But yeah, it would be sweet to like develop some new camera system to try to capture that.
Yeah, because I was thinking, I don't know if we're gonna do that specifically. It kind of depends on like.
Almost ready, Linda?
Yeah, 1 minute.
Depends on kind of like how the rest of the development goes. But I was thinking, I was like, it'd be really nice because
There we go.
Yeah.
And we got sound.
other people around here knew what they're doing. Like these cameras, like you're like, what's happening?
Yeah.
Or like, you have to come either using like some technology to like set it places.
Yeah. I know there's some folks in Florida that have used like camera traps to get moths pollinating ghost orchids.
Okay, everyone. Good evening. My name is Mark Broman. I'm the Executive Director of the Wachiska Audubon. Wachiska is an independent chapter of the National Audubon with a geographical area covering 17 counties in southeast Nebraska. You can follow us on our website and also on Facebook. I was out at Prairie Pines almost all day today. We've almost completed our transition. We've got the internet's up out there, and on Monday we should have the phones transferred, so we'll be almost 100% out there by next Monday. So be sure and stop by and see us. They're open every Friday and Saturday from 9 to 5 for the general public, but when I'm out there, I'll probably have the gate open and we'll have some folks stopping in. So if you're out in that part of the world, 112th and Adams. So if you want to come out, it was beautiful today. It was snow covering everywhere. There was deer tracks, coyote tracks, squirrels. So it was quite something. And then we had a lot of birds coming into the bird feeders. So it was pretty nice to look out the window and see that. We have hired a new office assistant. Her name is Wendy Bantam, and she'll start next week. She's been in a couple times and worked with Arliss. We'll have some transition times. And at the next month's meeting, we'll have a reception for Arliss after our meeting. So next week or next month, we'll celebrate all the good years that Arliss has put in with Wachiska, and we hope that we'll continue to have her help in the future. She just won't be coming in to work out at Prairie Pines, but we hope to continue having her help us out on some projects. I've got some announcements on a couple things coming up. The first thing, on March 20th, right here at the church, the Unitarian Church at 7 p.m., we're going to have an environmental candidate forum, and that is all the city council candidates. We're hoping to get both parties, but right now it looks like the Democrats are signing up and the Republicans are kind of shying away, but we're hoping to get as many as we can. So that's March 20th at 7 p.m. here at the church. So if you want to come and hear what the city council candidates want to say about environmental issues, we'll have some prepared comments and then or questions, and then we'll allow the audience to ask questions. So that'll be March 20th. This Saturday, just coming up in 2 days, we're hosting Bird Encounter at the Isley Library from 10 o'clock until 1. That's located at 14th and Superior Streets. There will be several bird activities, including live raptors. Betsy Finch will be bringing in some probably hawks and owls. We'll be making pine cone bird feeders. There's a bird banding display for the kids. Stories, showing them how to use binoculars and much more. So we're really geared towards elementary school kids. So if you have kids or grandkids or have some neighbor kids, bring them on out to Isley on Saturday. It might be a little cool, but 10 o'clock to 1 o'clock, we'll be out there. And we've signed up to have a booth at Lincoln Earth Day, and that's April 19th, and it'll be from 10 to 2 at Innovation Campus, the old state fairgrounds, where it's been the last few years. We also have a booth set up for Return of the Thunderbirds on April 5th. From 10 to 3 at the Lincoln Indian Center, and I think we'll have all these things listed in the Babbling Brook too, so if you didn't catch them tonight. And then also watch the next Babbling Brook that will be coming out pretty soon, as we'll have the birdseed order, the spring birdseed order, so we're kind of scrambling to get that all together with the move and everything. If you don't get one or haven't seen one, get a hold of me and I will personally email you a copy of the order form so you can get those orders in. At this time, I'm going to ask any of the board members for Wachiska to stand up and be recognized. I know we've got a couple of people here, and I want everyone to be able to feel comfortable to approach them. So we've got a couple. We've got Teresa Pillar, our president. We've got Tim Knott up here. We've got Dakota and Ethan back there. So give them a hand for coming in and being here tonight and also serving. So if you have any questions about the board, they're the folks. But thanks again for your service. We'll take a couple minutes and see if there are any unusual bird sightings. I might start with Dakota and Ethan. I know they've been flying a single sandhill crane around Lincoln here the last week and a half or so, hanging out with some Canada geese. So Dakota or Ethan, let's get a microphone maybe, Linda. Do you have one ready? Or I can just repeat things maybe, but you got one handy?
Yep.
I know Dakota and Ethan have been out taking some pictures. I know that Teresa got some pictures of it also, but maybe Dakota or Ethan or both of you can kind of give us an update on what you've seen or what's going on with that crazy bird.
Yeah, we don't know much about it, but it seems to be hanging out with a group of Canada geese. A few have been banded. We can't tell the numbers on the bands, so we don't really know where those Canada geese are from. But it's pretty unusual to see a sandhill crane in the middle of Lincoln, an urban city that I don't know if that's been seen for like decades, maybe even.
Yeah, tell us where in Lincoln you guys have seen it.
Well, it first was spotted on East Campus along
Layton Street.
Layton
Avenue,
Street,
yep.
on the cornfields feeding with the geese, and then it was at the pond at the St. Elizabeth Hospital, and it flew over to the VA lawns across the street, and then seems to roost on in between 27th and Cornhusker Highway, right behind Super Saver, with the geese. So it's very fascinating to watch.
Yeah.
You know, and it's there sometimes, and
Yeah.
sometimes it's not. But it's been here for at least 3 weeks, I think. So.
Okay. Yeah, a friend of mine said they were out driving on 70th Street the other day, and there were all these people with these big lenses taking pictures across the lawn. They didn't know what the deal was. Well, that was when it was hanging out at the VA over there in St. Elizabeth. So yeah, let's go ahead and see other birds. Since Linda's got the mic, anybody else have? So we got one up front here. Linda's bringing a mic over to you. We've had complaints that people at home haven't been able to hear the bird sightings, so I'll either repeat or have you use the mic.
I saw 8 trumpeter swans north of Ashland.
Okay, they're nice, big,
There's
beautiful
more
white
than
birds.
that, but I just saw 8 at one time.
Okay, good. Anybody else? Where'd you say, north of
You said
Ashland?
north of Ashland?
Yep.
Okay.
Okay. Anybody else have anything today? I know at the feeders out at Prairie Pines, we had cardinals and blue jays and lots of juncos. But the nice color against that white snow, the blue and the red was pretty magnificent. Anybody else have anything to throw out? Well, we'll go ahead and move on. This is a reminder that tonight's presentation and past presentations are available on YouTube. So if you like tonight's presentation and want to tell your friends, or you want to go back and see an old one or one that you might have missed, be sure and check them out. Tonight's my pleasure to introduce tonight's presenter. It's Dr. Catherine Hogan. Catherine recently joined Lawrence and Gardens in Omaha as the director of a growing plant conservation program, is the director of the plant conservation program. Since 2009, she's worked in native plants and habitat conservation and restoration, from New England wetlands to the Chihuahuan Desert to the open expanses of the Nebraska sandhills. Catherine enjoys long walks with her dog and partner in the prairies, and watercolor painting and cooking without recipes. So she's a risk taker. Catherine tonight's going to talk about the common and rare diverse faces of native plant conservation, and she'll answer questions at the end. We'll bring the microphone around so people at home can hear the questions. So with that, Catherine, thank you for coming tonight.
All right, is this loud enough? Okay. Hi, everyone. My name is Catherine Hogan. Thank you for having me here to talk to you today. Full disclosure, I'm used to presenting this with notes, so this may end up being a shorter presentation than normal because I might forget things if I can't find them on my document of notes here. So let me see. So I realized that I sent you guys the exact same information for my intro as I put on the slide, so I'm just going to skip that when we get to there, but I'm going to skip the slide with a bit about me. And then the first thing I'm going to talk about is a little bit of just kind of one piece of my dissertation research, which was here at University of Nebraska-Lincoln, and it was kind of looking at stability and synchrony in grassland plant communities. So basically, how do the plants kind of act all together over time? And then the second part is going to be a little bit about my postdoc research, which I just wrapped up before I came here, and that's going to be kind of looking at similar questions of plant conservation, plant prairie restoration from a very different perspective, a more basic science as opposed to applied science. And then I'm going to wrap up with kind of a little bit of an introduction or maybe reintroduction to the plant conservation program at Lawrence and Gardens and kind of some of our objectives and goals going forward and the kinds of cool things we're hoping to do in Nebraska and the Great Plains. So like I said, I'm going to skip some of this. That's a picture of me. That was the earliest picture I could find of me doing conservation work. That's in 2011 in Vermont when I was working at a wetland restoration nursery. I'm really good at taking photos of plants, not so much of people, including myself. So yeah, I got my PhD at UNL in 2022. And that's a picture of my dog up in the sandhills at McKelvey. And after that, I did a postdoc in prairie ecosystem restoration at Northern Illinois University in Northern Illinois, just west of Chicago. And then I joined Lauritsen just a few months ago, and I'm really happy to be back here in Nebraska and tell you a little bit about kind of what I've been doing as a plant conservation scientist. All right, so part one. Synchrony in grassland community assembly, or why are plant communities so weird? I think most people that have spent a lot of time with prairies know how much they can surprise us year after year, after even when we've spent decades with them, or in my case, not decades yet, but years. And this was a question that I actually started out my dissertation talk with, kind of just like a rhetorical question for people to keep in the back of their minds, is what purpose does variability or community change or community synchrony or asynchrony over time, what does that serve in terms of the grander ecological scheme? Like what function does it give to our prairies? And that's a picture of my dissertation research site out at Bobcat Prairie. So a more sciencey way to word the question that I was looking at for this part of my dissertation was how does community assembly, when I say community assembly, I just mean change in plant communities, patterns compare in restored or reconstructed prairies and remnant prairies, or prairies that have never been plowed. So to do this, I looked at a whole bunch of remnant and restored sites from south central Nebraska on two different preserves, the Platte River Prairies Preserve, owned by the Nature Conservancy, and the Crane Trust. And I'm going to keep the technical information in this presentation to a minimum, but just so you kind of have an idea of the kinds of data I was working with. I did not collect this data. I was given permission to use this data as part of my dissertation research by Ray Powers, who some of you may know. She now works for Xerxes here in Lincoln. And she did this part of work, and she was able to look at a little piece of the data for her master's project, but wasn't able to look at most of it. So I was able to kind of take it and look at some bigger picture trends. So what she did is she took 15 remnant unplowed sites and 15 restored sites for a total of 30 sites, revisited the same kind of roughly 20 permanent subplots every year. She went back there for 5 years. And the data she collected that we'll be talking about tonight is species richness, so just the number of plant species rooted within each subplot, and then their general kind of abundance in terms of a measure of percent cover within the quadrat. So the reason I start with this slide is because I think this kind of encapsulates a lot of the anxiety that restoration practitioners feel. This is the average species richness, so the number of species you'd find at any given point in a prairie averaged over 5 years for all of the remnants on the left and all of the restorations on the right. And as you can see, they're kind of all over the place, and that's kind of what we see in restoration and prairie management a lot. Is this is the kind of thing, if you're a practitioner or you're a researcher, you go out onto a prairie, and that's what you're going to notice is this drastic variability, and just sit there thinking, oh my gosh, what did we do wrong? What should we be doing? Like, where did all of this variability come from? What's going on? And the other reason that I like to start with this slide is that this is actually kind of indicative of one of the main results of this part of my dissertation was that in terms of the species richness level, in terms of just how many species you find in these prairies, there was essentially no difference between the restored prairies and the remnant prairies. I think it was less than one species per square meter that you would find different in terms of in remnants versus restorations. They're almost identical in terms of the number of plant species they support. So, but the other important piece to add to that, though, is okay, they have the same number of species, but do they have the same communities? And I'm guessing most of us here know what the answer is going to be to that, in the sense that no, they're not the same. They have very distinct plant communities. So all of these dots, the triangles are restored sites, these circles are remnant sites, and that's basically kind of a statistical summary of the plant community over that entire 5 years. So in terms of what species that were there and how abundant they were. You can kind of see there's kind of some overlap in terms of there's some restorations that are similar to a couple of the other remnants, but overall they're pretty distinct. The fact that those circles don't overlap that much means that you're finding different plant species and very different community patterns in restored species, or restored prairies as opposed to remnant prairies. So the same number of species, but very different species.
So did the restored ones evolve to have different species, or I guess if you planted different species, they would be different, but did it evolve away from what it was seeded to, or?
So the question was, have restored and remnant prairies kind of evolved to be different in terms of, so yes, it is partly what was seeded, but it is also, there's a lot of species present in remnant prairies that we don't really know how to restore yet. We know how to restore all the common species, but there's a lot of very specialized species that you'll find in remnants that we currently don't really know how to get established in restorations, which is part of what I'll hopefully be doing at some point in Lordson. So yeah, so the two main things to keep in mind as we look at the rest of the data are they kind of support the overall number of species, but their communities are very distinct. So this is the one technical slide I'm going to walk you guys through, and because it was one of the main metrics that I used in my dissertation, but I'm going to walk you through it step by step. So this is the variance ratio. This is what I use to measure synchrony and asynchrony. And synchrony is simply how much do plants behave similarly, and asynchrony is how much do they behave in different ways from each other. So in other words, a very synchronous plant community, you see here it's got time on the x-axis and some metric of abundance on the y-axis. And when you see a variance ratio of greater than 1, all that means is that species tend to decline and increase together. So they're kind of following the same abundance patterns over time. And then if you have a variance ratio of less than 1, it means the opposite. Obviously, this is a very extreme example for the sake of illustration, but asynchronous community means that as some species increase, there's a lot of species that tend to decrease, and vice versa in terms of their abundance over time. And then kind of the middle part of that is if you have a variance ratio of 1, that basically means it's random. There's no real pattern. So if that kind of made your eyes glaze over a little bit, as it did for me when I was first learning it, the main thing to remember is a variance ratio of more than 1 means species behave similarly. Variance ratio of less than 1 means they behave differently. Variance ratio of 1 means it's random. They don't really kind of respond to each other per se in any sort of perceived pattern. And if you guys are wondering the context of that, I walked into a huge patch of tick clover, and I was covered in seeds, and I was sad, and my laundry was sad for like 6 months, and everyone was sad. So that's the context of that picture. Okay, so what? So I just kind of just spent a whole minute and a half of this presentation kind of describing this one metric. Why is this important for people that care about prairies and are restoring prairies? So this kind of overlaps a little bit with what I'm going to talk about in the next part and when I talk about my postdoc research. But these changes in communities, so when you have species kind of declining and increasing either together or separately, this can impact their ecological function. So probably the simplest example of ecological function is just how much biomass these plants produce, just how big are they getting every year. And in the case of, say, a drought, if you have a drought that impacts plant communities and they all tend to respond in the same way, since, say, they all decline in abundance, that means there's going to be less biomass, which is less forage, less wildlife habitat, and that overall function is going to decline. Whereas asynchrony is kind of one of these main hypotheses where if you have species in a community that are responding differently to the same disturbance, then that can kind of buffer that function. Maybe you won't see such a decline in biomass and plant productivity, and that can therefore be a potentially really important component of prairie resilience, especially in the face of like climate change. So kind of my general question that I wanted to look into with this data was, do these synchrony or asynchrony patterns vary by plant functional groups? So a functional group is just kind of arbitrarily decided, not really arbitrary, but a group of plants like your grasses, your forbs, your trees, your nitrogen fixers. Those are examples of functional groups. But the one that I was specifically interested in is their lifespan. So are they annual, are they biannual, or are they perennial? And my hypothesis was that since annual and biennial, they tend to be very opportunistic, you could kind of say weedy species, I guess, they tend to be able to respond much more quickly to patterns like annual weather and whether or not it's a drought or not. So my hypothesis was that short-lived species were going to behave more asynchronously than perennial groups because of that kind of difference in their life history and their life strategies. So here's the main results page. So again, that 1 is kind of, it's a little bit light on this slide, but there's a dotted line that's 1. So if you'll remember, that's if the estimated confidence interval of the data crosses that 1, it means the species are behaving randomly. It means they're not really declining or increasing in parallel or kind of in opposition to each other. So what I found is that statistically speaking, they're all just behaving randomly because all of those confidence intervals of the bars, they are the confidence intervals of the data, they all cross 1. So it means that our plant community patterns are not statistically different from random. But when you actually look at the mean variance ratio, if you look at the annual forbs, the annual grasses, and the biennial forbs on the top, you have variance ratios that are a little over 1 to a little less than 1, which was kind of what I was originally thinking I would see, is that these short-lived species were going to be very asynchronous. They were going to all kind of respond very quickly and very differently in different ways. And when you look at the variance ratio for the perennial groups, it's quite a bit higher in terms of the average. It's over 2 for the entire community, which is mostly composed of perennial species, and it's almost 2 for both the perennial forbs and the perennial grasses. So I was part right. I wasn't all right, but I was part right, potentially. But this is probably my most favorite result from this part of my dissertation, and I've never been so excited by a null statistical result. In other words, a result that means there isn't any difference in the data that I'm looking at. When you look at the overall variance ratio for restored grasslands and remnant grasslands, they're statistically very similar. They're the same. So essentially, what we've got is they're both synchronous overall, but they're synchronous to the same extent. And if you'll think back to this plot that we looked at, showing that they have very distinct species compositions, this means that you have very different suites of species that are present in remnants as opposed to restorations, but they're acting the same with regards to how they're responding to management or drought or any other form of disturbance, anything that might trigger a change in plant species abundance. So basically, if I joked with my advisor, he didn't think it was as funny as I did, that I wanted to just have my dissertation title be Grassland's Gonna Grassland, because in terms of all the other metrics I looked at, and this is kind of just one of many that I looked at, there was very little distinction between remnant grasslands and restored grasslands. Again, they're kind of distinct community compositions, but in terms of their overall community patterns over time, they're basically the same. So, let me see, how am I doing for time? Okay. So what does this mean, kind of zooming back out to the bigger scale, what does this mean for grassland resilience? So this is where I'm kind of stepping away from the statistics and I'm giving you guys my hot takes as someone who thought about this very, very hard for 5 years. High variability, I think, doesn't mean that restoration and prairie management fails. If we go out and we see, oh, this prairie is less diverse, oh, this prairie is more diverse, but it's really variable, and we see all these things that kind of stress us out because we want to be able to predictably restore restorations and restore prairies, I don't necessarily think that that variability is a bad thing. Because when you think about it, variability, and if you talk to a bunch of other folks, like a bunch of folks at the Nature Conservancy and other folks doing grassland conservation across the Great Plains and in the Midwest, a lot of them will say, a lot of these practitioners will say that having heterogeneity and having variability in your prairies is actually really good, because that means that the same disturbance, if you have a drought that sweeps through the region, you're going to have some prairies that are likely going to be able to respond differently because they are different themselves. So kind of my main take-home message from that part of my dissertation is that I think remnants and restorations should be viewed, they're obviously distinct in terms of their plant communities, but I think they can be parallel sources of diversity and resilience for our prairies. So part 2, prairie ecosystem restoration. Again, kind of similar questions, but from a very different standpoint. Or, why are plant communities so weird? Again. Okay, so this was an experiment that I joined. It was it was a large grant-funded experiment that I was hired on to kind of lead the data collection, lead the management, kind of lead a lot of the kind of mid-level management of the project. And the fun acronym they came up with before I joined the project, I did not come up with this, is community assembly and prairie ecosystem restoration. So if I say CAPER, when I'm not talking about the little like salty things you put on your food, I'm talking about this experiment. So they received the 2 PIs, the head researchers on this project that I worked under. They received like $500,000 from National Science Foundation to create this, this experiment. It started in 2021. Obviously, I didn't join then because I was still in the throes of my dissertation, but I joined in January 2023, right before we started really kind of collecting data. Because this experiment entailed essentially restoring a bunch of experimental prairie plots and letting them establish long enough to the point where we could actually collect meaningful data from them. And kind of the overall broad objective, there were about a bajillion sub-objectives and hypotheses in the grant proposal, but the kind of one sentence summary of it is, how does community assembly or change in plant species and their abundance impact ecosystem functions? So things like biomass production that I mentioned in the last section. And while I was part of the department, we had to do flash talks where we would give very brief summaries of what we were all researching. And my supervisor, Holly, she told me that we all had to write limericks. So I got to write a limerick about CAPER, and it was plants assembled, or did they really? Some tried and failed, some got unwieldy. We measure plant function, might just have dysfunction, but there's trends, maybe we're not silly. So that's kind of a summary of everything we've learned from the project so far. I can go home now. Okay, so the ecological basis for this presentation, to get a little bit more into kind of the history of this part of ecological research is, generally speaking, we know very few things are universal in ecology, but generally we know when you have more biodiversity, when you have more species, you generally have more ecosystem function. There's always going to be exceptions to the rule, but that kind of seems to happen more often than it doesn't. However, some of the research that we've done kind of runs into some difficulties or maybe some limitations in terms of taking those experimental results and applying them to actual like conservation or actual prairie restoration. And one of them is that most research, in the interest of kind of trying to reduce bias, uses random plant communities, which that's fine, and we can definitely learn really interesting things from that. But most community assembly and community change isn't actually random. Plant communities aren't really random. There are some kind of rules and general structures they follow. So kind of to get at that, CAPER was based off of real-world prairie data, which we don't know of. I don't know of any other biodiversity ecosystem function type of like similar experiment that has done that. I could be wrong. But I'll talk a little bit more about what data they based it on in a minute. The other part of it is, so in that biodiversity ecosystem function relationship where we know more species usually equals more ecosystem function, we know that the number of species is important, but what about species identity and the individual characteristics and traits of those species? Those we hypothesize are probably also important, but the research isn't nearly as developed. So when I say functional diversity, I'm going to go into it more in the next slide. It's basically just the characteristics and the traits of plants. So do they have little thick leaves? Do they have big skinny leaves? What kinds of roots do they have? Are they annuals or are they perennials? Different things like that. So CAPER kind of attempted to incorporate all of these elements to try to fill in some of the gaps of the research that's been done by other similar experiments. So because scientists love to split hairs and define things 100 different ways, there are many different ways that you can define functional diversity, but the one that we used for this experiment is basically how distinct is any given species from the averaged characteristics of all of the other species in the community. So to give an example, this would be an example of, I would call a lower functional diversity community because we've got a bunch of grasses and we've got one flower and that's about it. They're all kind of fairly similar. If you compare it to a species that has, or a community that has the same number of species, but they're very different. Maybe you've got something from the rose family, you've got a grass, you've got a legume, you've got a sunflower. Those are all going to be functionally more diverse and functionally more different from each other. So kind of a human example of that is if we have a society like the one on the right that has an overabundance of plumbers and not much else, overall that community is probably going to be less functional than one that's really well balanced and has a bunch of different diverse professionals all filling different roles. So to kind of revisit, our overall objective for the project was to quantify this relationship, so the relationship between the biodiversity and ecosystem function of these prairies, to compare realistic plant communities with different levels of species richness, so number of species, and functional diversity, so again, those kind of traits of the species. And we looked at two different ecosystem functions. The first one is the one I already mentioned, the above ground biomass, so basically just how much live plant material do these plants put out every year at the peak biomass season in like late August. And then the second one is decomposition, or how fast these plant communities can essentially break down organic matter and put it back into the soil. So this one, so I said the experiment where most of our data collection took place was just at Northern Illinois University, which is a little bit west of Chicago. And it's kind of hard to see, but that's the satellite imagery of the experiment. And I'm gonna show you some more details about it on the next couple slides. But that's right on the edge of campus. And then the other location where we basically based these experimental plots on is Nachusa grasslands, which is a, yeah.
Do they have buffalo roam in this area?
At Nachusa, yes.
Yeah, do they roam on the plot?
So, I didn't have time to fit that into the presentation, but we actually did pretend to be bison and pretend to graze it. The question was if we had buffalo at both Nachusa and at the experiment, and we pretended to be bison. We did not bring in an actual bison, though we actually.
I know there's bison in Nachusa,
Yeah.
but did they actually graze on this plot at Nachusa that you had studied?
So as one of our treatments, we basically pretended we were bison and we simulated bison grazing on some of these plots. Yeah, but I did not have time to fit that into this presentation. There's a lot to this experiment that would not fit into this talk. But I'm happy to talk about it more later if people want to. Okay, so this is the kind of the overall experimental design. So this is just, if you remember the satellite imagery, this is the exact same thing, like we're looking down on it from above. Up is north. Let me see, there is 156 plots. And part of the reason why I'm showing you this is because I spent way too long figuring out how to make these maps, so I use them at every opportunity. And then, so we use 24 plant species. So basically, the lab that I was a part of, the evidence-based restoration lab, has for about 10 years been doing annual vegetation monitoring at Nachusa, where they basically go out and over about 200 different vegetation plots on all of the different restorations, they collect plant identity and kind of composition data, so we know what plants are doing in Nachusa pretty closely over the last 10 years. And we took all that data and we kind of summarized it all, and we picked the 24 plant species that occur most frequently. That they're some of the most abundant, 24 basically most abundant plants at Nachusa, and then we use those to create the different experimental communities for this. So this shows the species richness levels. The orange is 8 species, the pink is 12 species, and the purple is 16 species. And then functional diversity, which is essentially the same colors, only they're kind of randomized, well, not fully randomized, but they're kind of mixed up a little bit, where in the case of, say, plot number 1, I don't know if this has a pointer. So in plot number 1, you can see it's a mid-diversity, but if you look back at this, it has 16 species. So for any given plot, it's going to have a lot of species or fewer species, and it's going to have either not a diverse community or a less diverse community versus more diverse communities. And then I put in a photo timeline, which Sam, the first lab tech for the project, she took a bunch of photos. That's right after it was planted, or a little bit after it was planted in 2021. And that's its first fall. Kind of sad looking, the first winter. But then next May, it just takes off. And this is a little after I joined in May. And that is the second, or its third summer. This year we started doing a lot of the data collection. So you can't really see it in this picture, but I'm 5'1", and a lot of those plots are taller than me. Most of these, the silphiums and your big asters, were probably 7 to 8 feet tall by the end of the summer. So our first experiment that I want to talk about is the decomposition experiment. So basically we took a piece of filter paper, which is essentially 100% cellulose. It's the same kind of material that makes up plant tissues. and we put it in a mesh plastic bag, and we staple it so it's right in contact with the ground. We walk away and we leave it for the better part of a year, and we kind of come back and start checking them the next spring. And when it looks like some of them are starting to disappear completely, we pull them all and we basically get a comparison weight, which I should have mentioned before we staple them to the ground, we get a really, really precise, like to the 10,000th of a gram weight for each of those pieces of filter paper. And then we weigh them after we pull them out of the field, so we can basically calculate what percentage of their weight was lost to decomposition in that time, after we very, very gingerly clean all the dirt off them because they get very dirty sitting out in the field. And what we found is super noisy data, not surprisingly, but there may be a little bit of a signal in terms of how functional diversity affects decomposition rates. And the thing to notice about this plot is I flipped the y-axis. So this is 0% weight loss. So the lower down the dot is, the more weight was lost. So we had one that disappeared completely. We lost it, couldn't find it. It was presumably completely decomposed. But most of them you can see are quite a bit higher up. And the average, I don't remember the numbers off the top of my head, but the average total decomposition for the 3 different panels you see here is the mid-functional diversity actually decomposed the most, which was kind of surprising. We expected it was either going to be the lowest or the low functional diversity or the high functional diversity, followed by the low functional diversity, and then the high functional diversity actually decomposed the least. Now, we haven't really run like a full statistical analysis, and we're talking about a pretty small difference. We're talking maybe like a difference of 5 to 10%, which doesn't sound like a lot for this, but if you kind of blow it up to the scale of all of the organic matter sitting on a prairie, a difference of 5 or 10% could potentially be a pretty big difference in terms of how much organic matter is getting put back into the soil. So then the second one, and this was probably our biggest one in terms of the number of people we had, we went out to every plot. We visited all the plots. It was late August, kind of peak biomass season, and we clipped a standardized amount of biomass from each one of the plots, and we sorted it all to species, which, let me tell you, on a windy day, that is not fun, trying to sort out Poa pratensis from little bits of blue stem. It gets pretty fiddly. And then we take it all back to the lab, and we store it in bags and bags and bags and bags. And I don't think this is even all of them. I think we had more. We had stuffed in like every corner of like 3 different lab rooms. It was kind of ridiculous. So we would dry them all down till there was no moisture left or very, very little moisture left, and then we would weigh them all to see what's kind of the average weight of a species in a given plot, whether it's one of the high diversity plots or one of the low diversity plots. And what we found is trends, but again, not what we were expecting. So remember how I said when you generally have more species, you generally have more function? We saw the opposite. So instead, what we saw is for the orange plots, which are the low diversity, there's a lot more biomass, but it's also super variable. And with the high diversity, there's a lot less, but it's not variable at all. It's very consistent. And this is actually a different hypothesis that we weren't even thinking about. I say we, I wasn't part of the grant writing process, but that we weren't even thinking about when we planned this experiment, is the idea that high diversity systems also sometimes have more stable ecosystem function. They don't vary as much as low systems, so as low diversity systems. So kind of the reverse of what we were expecting, but also super interesting. So I don't know what the actual final result of that's going to be. This is the first year of data. I didn't have time to combine it with the second year, but the trends across the 2 years are basically the same, which is really good because it shows we had a good method and there wasn't something screwy with our data, but it didn't really change the results much when we added a bunch more data in. So let me see, what time is it? Okay. So to kind of go back to that limerick, plants assembled, or did they really? Some tried and failed, some got unwieldy. So we really tried very hard to design caper to be realistic, like we really did. But on top of the irregularities and kind of the weird, like, wonder what like caused that in the data, some other irregularities we noticed pretty early on were that the plants at the plots were really, really big compared to the ones at Nachusa, like way bigger. So we decided, well, we've got this great experiment set up. Let's collect some data and like see what's up. So we decided we wanted to compare functional traits, so the characteristics of individual species at both caper and Nachusa. And Anna, who is our rockstar REU intern that joined the lab, she took this on as part of kind of her internship project for the summer. So she went out and picked 2 plant species that we thought were like, okay, these are really weird. Like these guys are like, they're like a bush compared to what we see out of the prairie. Not at all growing the way we would want to see them growing in an actual prairie. So we called these the Frankenplants. And then we picked 2 that it's like, okay, they're still kind of weird, but like they're closer. So like, but they seemed like they're a little bit more normal. So these were our kind of well-behaved plants, as good of a control as we could get. And then she went out and she measured several different functional traits, several different characteristics of each plant at both sites. So this isn't really a great comparison because we're comparing, we've got pictures without flowers and with flowers on the top, so that's kind of not a great comparison, but you get the idea. Basically, the ones at Caper on the top were really big. She's taller than me, she's probably 5'6", so you've got this stiff goldenrod that's almost 6 foot tall, versus at Nachusa, it's just like a single little leaf kind of coming out of the ground. And sometimes you don't even really need statistical tests to be like, oh yeah, they're all Frankenplants. They're all kind of, this is just plant height as one example of one of the traits she looked at. And even when you corrected for like some plants at Caper were blooming, so we tried to get ones that weren't blooming, which is a better comparison to most of the ones at Nachusa, which were also not blooming. It's a very dramatic difference in terms of their size. And their other traits were also equally just as weird. I just didn't want to overwhelm you guys with all the plots in the world. Okay, so what? Like, what does that mean? So because we know that the caper is not really, quote, as realistic as we wanted, does that mean it's a bust? Does that mean we failed? No, I would think that the value of these really small experiments like the one Anna did really help us to quantify that difference. So in terms of, it's one thing to be like, oh, well, they're different, so I guess we can't compare them. But if you can look at a bunch of data from a bunch of different plants and it's like, oh, we can actually quantify the difference between the experiment and the real world, you can kind of sometimes still figure out how to translate it. Be like, okay, you know, maybe it is going to be a larger effect at Caper, but if you have comparative data at Nachusa that shows the same patterns, maybe they won't be as extreme, but the patterns could still translate. So I'm trying to think if I had any other, like, no, I don't really think I had any wise words to wrap up that. But it was a really awesome project, a really awesome team. Really makes a difference when you're out there and it's like 95 degrees and 95% humidity when you have great people to spend time with. So okay, the last part here, I think I've got time here. Plant conservation at Lauritsen Gardens. So this is gonna kind of feel like a big topic switch, but it kind of represents, it's been quite a change for me going from being a plant community ecologist and thinking about communities with dozens to hundreds of species to thinking about not only species, but the rarest of the rare, like orchids, which are really weird, guys. Like the things I've learned about orchids that I never thought I would be learning as a community ecologist have blown my mind, but I'm thoroughly enjoying it. So one of the reasons I was so excited to get this job at Lauritsen and to come back to Nebraska is now, especially having worked in prairies in Illinois and as well as Nebraska, I can say definitively that conservation in Nebraska is way cooler than conservation in Illinois. And I say that with no shade to Nachusa and the eastern tallgrass prairie. It is absolutely beautiful, it is diverse, but Nebraska is so variable in terms of you have this massive expanse from the southeastern side where you pick up all of the eastern species that hit the Missouri River but can't get further west, and you have the western species in the panhandle, and you have southern plain species coming up from the top, and the species kind of like even like glacial remnants that are coming down here that have really unique plant communities. So really what I've been telling everyone is doing conservation in Nebraska on these rare species is you feel like you're at the edge of everything. Like we don't really have many endemic species. We have very few species here that aren't found anywhere else, but we get a little bit of the eastern species, we get the western species, and because we have all of those edge of range species, it's quite possible, and this is one of the things I'm hoping to kind of look into down the road, that the plant communities we have of these rare species are actually potentially genetically very valuable to the conservation of the species because they live at the edge of the range. They are the most likely to be the ones that are having to actively adapt to new and inhospitable conditions. So that was kind of something that I didn't really realize as I was just working more on the communities that I'm starting to learn now that I've kind of started focusing on more specific species. So some of the, I think I was talking to a couple of folks earlier, and I know Jim has come and spoken. Jim Locklear, the previous director of conservation at Lauritsen. So this is maybe kind of a reintroduction to the program for some of you, but I just wanted to kind of go over some of the main goals, which are kind of similar to what Jim was focusing on with his work here, with a couple differences. So obviously, to conserve these plants, we need to know where they are, and we need to know which ones are the most endangered. So a lot of what we're going to be doing is doing conservation assessments, trying to find populations of these rare plants and kind of assess how healthy they are they actively declining, are they stable, things like that. And then once we kind of determine where some of these species of interest are, it's a matter of kind of responsibly collecting those plants' genetics, usually in the form of seeds, though not always. And so the third one is conduct applied research on exceptional plant propagation restoration. By exceptional, I mean species that have very, very specific life history and habitat requirements. So kind of the stereotype of that is our orchids. Orchids are very specialized, super highly specialized, super high, high demand, I guess you could say, in terms of like the kinds of conditions that they need over time to thrive, and they're never going to be super common, or almost never. They're almost always going to be fairly rare and fairly scattered. So it's going to be conducting applied research to figure out, okay, how can we germinate or how can we store these seeds, things like that. Yeah?
Besides orchid, what other species are imperiled?
Oh, so many. There's over 1,500 plant species in the state of Nebraska. So the current list of species that are considered of conservation need, that list is currently getting revised by Nebraska Game and Parks, but it's currently at over 400 species. So that includes a smattering of everything. A lot of them are like these kind of really specific species, like our orchids. There's also a couple moonworts, like a little kind of ferns that are super rare and just occur in a few known locations. But there's also one, like not the one in this picture, but there's a species of milkweed, Asclepias lanuginosa, the woolly milkweed, which is our earliest blooming native milkweed, and it's almost never found in fruit, and it's almost never found with actual like viable seed on it. So it's kind of all over the place. The orchids are, I think, overrepresented because they're so specialized, but there's just about every plant family that's found in Nebraska has some imperiled species in it. So then a lot of what we do obviously is not going to work if the public doesn't have an awareness or value our native plants and ecosystems. So outreach is a really important component of what we do. That's going to be working a lot with people, the other, like the horticulture folks at Lauritsen Gardens that are working to kind of incorporate more native, kind of more ecologically appropriate species in the displays. And then last but not least, we're mostly going to be focusing on Great Plains species, but we do have a small collection of orchids, mostly from Madagascar, that we inherited from the Dorley Zoo, and we're going to be working with them going forward. And that's what I'm going to share a little bit about now, because I started in November, obviously haven't had a chance to get out in the field too much yet. So, and this is also kind of to just draw attention to the fact of how well situated Lauritsen is to do this kind of work. That map on the left is kind of a rough outline of the Great Plains. And if you lay that over this other map on the right, which is Botanical Gardens doing plant conservation, there's not much going on in the Great Plains. We've got there's Mobot down here in Missouri, Botanical Gardens, Denver over here. We've got Morton Arboretum and Chicago Botanical Gardens over there, one in Minnesota. But there's really not that much going on otherwise in the Great Plains in terms of organizations working on plant conservation. So Lauritsen is really well situated to kind of be a world-class kind of garden of the Great Plains in terms of plant conservation and paying attention to the plight of our native species. And some of you may not know this, we have brand new plant conservation facilities. So towards the back of the garden, there's a new plant production center, and the conservation lab, where I'm going to be showing you some pictures from next, is up on the second floor. It's not accessible to the public, but I'm going to give you guys a bit of a photo tour to show you kind of what we've been working on. So this is the lab. We named it the Jim Locklear Conservation Lab in honor of Jim, the previous director of conservation. It still kind of get filled in with we'd need to get some more equipment, some more freezers. But the one thing that we've got here this is really special is this clean room over on the side, which is where we keep all of our, we call them in vitro plant containers, where they're basically plants that are being grown in sterile environments. Most of these are Madagascar orchids. We have a couple other species from Bermuda and Borneo and a couple other mostly tropical locations. But basically we're working with the genetics of species that were collected by by Marge Frum. She was the plant conservation scientist at the Dorley Zoo before they discontinued their program in 2020. Lauritsen basically inherited all of her work and all of these orchids. And we're kind of using them kind of like the zoo uses animals, hopefully to treat them as kind of ambassador species to kind of show people some of the diversity of plants from across the globe that are highly endangered. Most of these orchids that we're working with are endemic to a very small part of Madagascar, so they're not found anywhere else on Earth naturally. And most of them are very little known, and so if we don't really know that much about them, we can't really even make sure they don't go extinct effectively. And that's just a very satisfying picture after we did inventory and everything was very tidy. And some more fun pictures. This is what I mean about orchids being weird. That's what they look like when they're germinating. Whoops. Orchids, they don't have kind of like a typical seed like most plants. It's basically just like a naked embryo. And they only germinate in nature with the help of fungal partners. So if we don't have fungal partners, which we don't even know the fungal partners that most of these orchids have, so we can hardly use them, we kind of have to play the part of the fungus and do a bunch of other things in terms of keeping them in sterile conditions until they can germinate, and then they're put in these little containers in sterile conditions in kind of a nutrient-rich growing medium, and we kind of keep them there until they're big enough to grow on their own and photosynthesize on their own and produce their own food without those fungal partners. That's just an example of one other species we're working with. It's a species that is endemic to seaside caves on Bermuda and nowhere else. So it's one of those situations where it's this extremely rare specific species that we're working with in vitro conditions. So looking forward, like I said, we're going to be kind of learning from this current collection, learning how to work with these orchids, how to keep them healthy, how to grow them. We're also going to be surveying rare plants in a few different locations across Nebraska. Some of that's going to be what we call conventional seeds, so it's going to be basically the seeds from species that you can dry them and throw them in a freezer, and they're good for decades. And then we're also going to be working with these more specialized species, like some of the orchids, and kind of trying to figure out, like, okay, can we get these seeds to germinate? Can we do some storage trials to see, like, what kind of storage maintains the viability of the seed, and kind of those kinds of questions. And then a lot of what we do is also going to just be building partnerships. Nebraska is a really great place to do this kind of conservation work because we're really good at working with each other, I think, and conservation can't happen and be effective in a vacuum. So a lot of what I do is just kind of building those partnerships with people. And then this is I just want to highlight, this is one of the species. I am pretty certain at this point we're going to be able to do some surveying of a couple different populations. And this is one of those species that I mentioned. It has a pretty wide range across the eastern United States, but it kind of hits the Missouri River and it doesn't get much further than that. So we have these kind of few isolated populations on this side of the river that may have potentially very unique genetics compared to the rest of the species. And then this is just a list again of some of the partners we're working with. Really quickly as we're wrapping up here, the one I wanted to highlight is the North American Orchid Conservation Center. It's a very small but mighty bridging organization that is working to kind of standardize and kind of collaborate across institutions with basically everyone who's doing North American orchid conservation work. So we're going to be contributing some seeds and leaves and roots to a collection they have of orchids across North America to try to kind of isolate things like what fungal partners do they have, what's the DNA of the populations, and how can we preserve the seeds. So we're going to be working with them kind of in tandem, working on the same things to hopefully conserve some of these super rare orchids we've got. Well, that's it. Thank you.
We'll take questions. Remember, raise your hand. I'll bring the microphone around. We'll have a microphone on each side of the room. So raise your hand and I'll give you the microphone so we can get them online so the people at home can hear the questions.
And Jala Foley, a new
Well,
person
we'll start
to
here since I'm close.
Wachiska, will be running the microphone around to this side of the room.
Okay, we got a question here.
So what resulted in those Franken plants? Was it water or?
Oh yeah, see, I told you I was going to forget things. So our current hypothesis about the Franken plants is the restoration method. So most restorations are seeded. So you're just taking these high diversity mixes and you're broadcasting it on the surface. To get the very specific plant communities we needed for the experimental design, we had to use plugs. So we had to basically start with already started little plants that had good root systems. So we think what happened is we gave all the plants that we planted a head start kind of inadvertently because we needed them to survive or we wouldn't have had an effective experimental design. So they got babied and they kind of started with a good rootstock and they didn't really have to compete at the seedling stage like plants at Nachusa do. So that's kind of we haven't really looked into that next yet, but that's kind of our running hypothesis as to what happened.
When you studied the functionality of prairies, did you ever look at populations of insects or birds or anything like that as far as and how diversity would affect the function of that?
No, we didn't look for this study specifically. I know a lot of people have, particularly in prairies, and it's kind of the same in the sense that the more diversity you have, it might not necessarily, depending on kind of when you go out and count the bees or how you count the bees, it might not lead to more bees, but it usually leads to better established and more stable communities. So it's kind of what we were seeing with like the biomass production, where a more diverse community doesn't necessarily have more function, but it's a lot less variable. So it's kind of more consistent in terms of the function that it might provide over time.
Okay, additional questions? Got one here, Gerald. I'll grab them since I'm close.
I was curious how you kept those plots so clean of annual weeds. Was that all handwork or did
Yes.
Okay.
A lot of work by Sam, the lab tech, and Anna, and a bunch of volunteers, and me, and just about anyone we could rope into it.
Okay.
Yep.
I was kind of wondering when you were talking about those plants being so much bigger, if it was that ground cloth between them, if that warmed the soil more, and then whenever it rained, it concentrated the water in that zone where the plants were, it rained,
Yeah.
and they didn't have to compete so much for moisture.
I think that might have been part of it. I know one thing for the data that we collected, we kind of stayed away from collecting any sort of data right around the edge of the plot. We tried to kind of get away from the edge because that is a documented thing that happens in these experiments is you get what are called edge effects. So the plants that are growing on the edge of the plot, the plants that are growing on the edge of the experiment, they will tend to sometimes react a little bit differently because of things like that. So we tried to avoid that. Don't know if it was 100% successful, but yeah, that is something that happens.
Any other questions?
So here in Lincoln, we've had installations of prairie strips in a lot of our parks. And I noticed that the plant population seems to have changed dramatically within about 2 or 3 years. In other words, we started with a lot of flowers and now it's all grass. So is that exactly what you expect? Or does that figure in any of the work you're doing? Or?
Yeah, not to really the work that I talked about here. Another chapter of my dissertation looked at that a little bit, and that is kind of something that's seen a lot is the the grasses are really great and they're a really important part of prairies, but they're more competitive over time than flowers. So depending on the management, that is what we see happen a lot, is you have this initial influx of kind of the flowers and even like weedy volunteer species that come in, and then it kind of drops off over time if it's not managed appropriately, which is really hard to do in these kind of like controlled like city situations, obviously, where they're not just gonna go lighting like roadside strips of prairie on fire. I mean, I guess they could theoretically, but I think some people would take issue with that. But like ideally, you use management to kind of prevent that from happening. But without management, that is usually what tends to happen over time, is the grasses will outcompete the forests.
Okay, so in a restored prairie, like you were considering at some points, is it considered okay or good or bad?
To have more grasses?
No, to have it all go to grass, which is what seems to be happening here.
Yeah. From a diversity perspective and from the perspective of a lot of organizations I work with that work in conservation, not ideal. It's not necessarily bad, like having a lot of grass isn't necessarily bad, but when that's the only thing you have, and that's overall time, and that's all of your different prairies that you're managing, if that's the only thing that's there, then that can become an issue. But like there's some wildlife species that they need that really thick cover, and they need that really high dense grass. So the main thing is to kind of keep what's called, there's a bunch of terms that people use for it, but kind of keep a variety, like okay, it's okay to let this part get a little bit tall and grassy, but this part we're gonna burn it and we're gonna graze it and we're gonna kind of suppress the grasses a little bit. So it's the kind of thing where it's okay in moderation, like most things, but when that's all you have, it can be problematic.
Oh, okay, I'll push it one more step.
Yeah.
I have a whole bunch of milkweed pods, and I've had to restrain myself from going over to these plots and putting the milkweed pods out so that we can get some flowers back in there. So would that be recommended or not recommended?
Oh man, now I'm just having to step into kind of the guerrilla gardening scene. I mean, I don't know what the city would have to say about it.
Well, that's why I haven't done it, because it's not my plot. I'm not in charge.
I know in another prairie, if the owners didn't care, some milkweeds are kind of weedy, and they can kind of compete with grasses, but like some can't. So might work, might not.
Thank you.
How about one more question for Catherine? We've got one over here. And then we've got some refreshments out in the concourse. And then also be sure and sign in if you haven't when you leave. We like to keep track of everybody.
Our members are kind of familiar with the Western Prairie Fringed Orchid. You know, the real frilly one that is so beautiful. And I mean, I'm sure you all know about that one. I mean, you're very familiar with it. Mm-hmm. But it seems to have disappeared from places, and a lot of us probably think it's disappearing because of the dry conditions that we've had for several years. Is that happening to other orchid species in Nebraska?
So again, keep in mind that I've only started learning about orchids a few months ago, so I've got a lot of catch up to do. It might be the dry conditions, though some orchids, like there are native species of orchids that grow in the panhandle. So like there are some species that can handle fairly dry conditions. I think it's more likely kind of the management issue of, or it's probably multiple things, but I suspect a big part of it is management that leads to fairly homogeneous, similar plant communities over time, because some of these native orchids, they spend most of their life, like they can kind of go dormant underground for years, which is part of what makes it so hard to learn about them, because you can go back to the same population the year later after you just found hundreds of them and they didn't come up this year. Like they do stuff like that to kind of, as a way of dealing with short-term changes in the environment. But if 10 years they can't grow because there's always heavy like grass cover or woody encroachment, then like, yeah, over time that might kind of combine with the effects of drought, might be combining with some other things. So it's probably a lot of things going on. The drought and like drier conditions is probably part of it, would be my guess.
Well, thank you, Catherine, and I'm sure she'll stick around for a few questions afterwards, and thanks everyone for coming tonight.
Thank you.
Catherine. Be sure and stop out and have a cookie and sign in before you leave out there.